Hello everyone, my name is Peter Winter. I'm a taxonomist employed by SANBI and I'm based at the Compton Herbarium, which is at the Kirstenbosch Research Center. I've be talking about how you can go about getting to know Heliophila with some pointers on what features to look out for when trying to identify them. And I'll also just briefly touch on other members of the Brassicaceae that can be found in the Greater Cape Floristic region. These will include some indigenous species as well as the more common alien invasives that you are likely to encounter, and then just briefly say something about the threatened species. Before I get on to the rest of the presentation, I'd just like to acknowledge your input, people like yourselves who have contributed images that I've relied on heavily in this talk. And it's always been a dream of mine to be able to have this reiteration in engaging with other people while contributing to building knowledge around a group of plants. So Heliophila is in the mustard family or Brassicaceae. Currently there are 104 or more recognized species, mostly endemic to the Cape Floristic region. Since 2019, there have been 13 new or reinstated taxa, and at least another 13 have yet to be described. And citizen science has a huge potential to clear up the taxonomy, and I'll briefly talk about some of the taxonomic problems and where we are in the development of knowledge around this group, and with the aim of creating more order. So I think we at a stage where professionals and amateurs can work together in this ordering of our knowledge. As I said, I'll be talking about naturalized species and potentially threatened species as well. So the one example of the state of the taxonomy is one needs to realize that the last work, which was done in 1970, was in what we call the consolidation phase. So taxonomy goes through explorative phase, a consolidation phase, and currently we're in a refining phase. But the last work was done by Marais in 1970 and in the consolidation phase. So his approach was to look at mainly just at specimens, not rely on going into the field. Certainly it wasn't so easy to get beautiful images as it is today. So he wouldn't have seen the striking differences among populations. So he would be inclined for a small region like the Swartland to group things that shared certain characters together under one species. But now we can critically look at that group and also realize that not only did he include these three entities, but two other entities as far north as the Bokkerveld Plateau into this concept of Heliophila acuminata. On the left we have the core concept. This is the originally described Heliophila acuminata, and it is endemic to the southern part of the Swartland. This population is only known from the 24 Rivers area near Porterville, and a few populations like these are scattered from around Mariatsburg down to Riebeek Kasteel. And so it is clear now, looking at the variation, that there's room for reinstating some of these entities. You'll notice that these photographs were taken by Stephen Cousins. He found a population of these, and I helped him to identify what it was. and we embarked on a sub-project where we were going to reinstate or rename some of these entities. Then the second example is one of the better known names that has widely been applied to anything with a blue flower in the annual clade along the west coast, annual group of species common on the west coast, Heliophila coronopifolia. But you can see by these red circles that the samples that were used in this analysis came out in at least two distinct places. You can see this one is closest to Heliophila adpressa, which is a coastal lowland species, and this one is closer to two other species, Linoides and digitata. More about that now. This would to visualize what we're talking about. This is the one group that was closest to Heliophila linoides and digitata, up to 2 meter tall annuals, compared to a similar, also similar coloration in both leaf and flower, but only about 20 to 30 centimeters, so an order of magnitude difference in size. These ones often have hollow stems and occur in a different area from these ones. And then the proper Heliophila coronopifolia lacks the glaucous leaves, has these bright green leaves with many more lobes than these ones. And the nice thing about INAT is you can quickly visualize. Once people have contributed enough observations and they've been correctly identified, then it's very useful to visualize the distribution ranges. And so you could see that 2 meter tall entity is this bit encircled in yellow. And when I looked closer at this, it's quite striking how it correlates with the area in which rooibos tea is grown, in the sandveld. Sand fynbos has been cleared to make a lot of the rooibos tea plantations, and this is the habitat that is shared with this particular entity, which there is an older name for it, Heliophila zairi, and I plan to reinstate that name. Then the next one down is in the Langebaan granite strandveld. That also has representatives on the Cape Peninsula in the lowlands, and we're not sure if it's the same entity or whether that could be two separate species, but for now we're treating them as one. Then the next entity is endemic to the Swartland granite Renosterveld, these two areas here. And yeah, sorry I advanced too quickly, but you can get an idea that the distribution ranges of these things can be quite distinct, and so there's a lot of room to work on and in refining the taxonomy of this group. Now, to look at the genus as a whole, this is an extract, a combined extract of all SANBI records and the Global Biodiversity Information Facility, GBIF, records, combined together in a distribution. So this represents the total known distribution range of the genus. You can see up to the Brandberg here in Namibia, and up as far as Limpopo in South Africa. And this gives you an idea of so where some of the species in the less species diverse parts of the country, you can make out one or two species. This is Heliophila rigidiuscula, grassland species. And then along the coast, these blue dots represent Heliophila subulata, often photographed, and then the smarty box here is the real diversity in the Western Cape, centered around the Cedarburg and the West Coast mainly. But if you look in your area in Southern Cape, there's also quite a lot of diversity. And then we go to INAT, and you can see we've only been busy with INAT for in the order of 10 years now, and already we've got quite a good coverage. So it's a fair match for the total coverage that we saw in the previous slide. As you can see from the figures here, this was a year ago. It was at 3,200 observations. It's currently at more than 4,000. Then it was 69 species represented. I think now it stands at 73. So in a year, 4 new species have been added. So you can appreciate that as time goes on, it'll be more and more difficult to get the lesser known and the rarer species. And there's still a long way to go before we get to species number 104 or species number 120, when all of the undescribed ones have been added. INAT is very useful in terms of providing excellent population coverage. This is Heliophila subulata with the most observations. These were those blue dots in one of the previous slides. And the beautiful thing about iNaturalist is, without having to go on a field trip, you can get a good overview of the variation just in a whole lot of images from all these different areas. Not only is it in terms of populations, but in terms of area. This is one of the more widespread species, Heliophila carnosa, all the way from Namibia and up to the Witwatersrand, mainly along the escarpment, but extensive variation in the Western Cape as well. So again, without having to travel to each of these populations, one could get a fair idea of the variation. Then INAT is also very useful in terms of new range records that have been elucidated. The type locality of Heliophila trifurca is on the Upper Karoo, the southern part of the Upper Karoo, but it's better known from the northern parts and into Namibia, especially the Bushmanland area. And surprisingly, there have been virtually no records in the Great Karoo or in the Little Karoo area. Then only in the last few years did we get some records from the Gamkapoort area, and then a lot, because there's a lot of INAT activity in the Southern Cape. We've quickly collected a lot more range records in this area. So INAT is very useful when it comes to extending the known distribution ranges. Okay, now what characters are useful? You can see the range of diversity in fruits in this genus. It's quite staggering. And yeah, not only in the shapes, long and narrow, also in the way the seeds either bulge or don't bulge. This one has a very flat side, whereas this one is bulged by the seeds laterally as well as front and back. So there's variation in width. There's variation in the remnant of the style that tops the fruit, the cilique as we call it. Then there's shorter ciliques down to little ones like this, which are actually wider than they are long, and when they become like this, we refer to them as silicles. This would also be a silicle, quite broad and flat, with just 2 or 3 large winged seeds in them. You can see how with the narrow wastes between the seeds in these. So this gives a very useful character to identify this species. Going to the flower diagram of Brassicaceae, getting a little bit more technical. This is like looking from the front of the flower, and with the sepals being the outer whorl, and the petals, 4 of each, 4 sepals, 4 petals, and then instead you would expect 4 filaments, but instead we have 2 sets of filaments. The outer filaments, there's only 2, and they alternate with the petals. So the whorls normally alternate going from out to in. So the petals alternate with the sepals. Then the next whorl of filaments alternates with the petals again. And the next whorl of filaments alternates with that first whorl. Now, what you can see, yeah, so what I want you to, this is the ovary in the middle, with a midline splitting the fruit into 2 halves, symmetric halves. So this is not quite yeah, it's a radial symmetry, but it can only be, there's only 2 symmetric axes, one going along the median line and one going along the lateral line. So I wanted you to concentrate on this area, on the inside of one of the sepals and on the inside of a filament, and the between area between the edges of 2 petals. So what happens is that some species have an appendage on these filaments that forms a little cover going out towards the sepal. Then likewise, each petal has a little appendage extending towards the middle and completing the formation of a little lid above this opening. And in this image you can see it more clear. There's the petals, the sepals here with the petals removed, and you can see the sepal actually forms a little pouch where nectar collects. There's a scanning electron micrograph of the base of one of the filaments, and you can see that little cover appendage extending from the base of the filament, and below that the nectary, which exudes nectar into this pouch. And these covers somehow seem to protect that resource, or for certain insects to act as a pollination reward, presumably. And here you can see the petal appendages, which likewise help in forming that lid to protect the nectar pouches. This is just a line drawing that one of the collectors of one of our specimens drew on the specimen he collected, which shows the appendages extending out towards the sepal and the pouch then together forming this nice little protected area for the nectar. So that the presence or absence of that is very important in identifying different species. Then on INAT, there's some traits that are not always apparent on herbarium specimens or in the literature descriptions, like the reflexed sepals and petals of Heliophila cornuta variety squamata. And apart from this species, it's only known in 1 or 2 others. And so it becomes very helpful to eliminate species that aren't relevant to the identification. And some other things to look out for are the color of the filaments and the buds, whether they are direct or held upward, erect, or whether they are reflexed. So here you could see these two share filaments that are purple, but yet they are in two different groups, and that you can only tell really by the reflexed pedicel in the bud stage. These two are actually in the same main group of Heliophila, and but they can be separated by the color of the filaments, and also in this case the filaments are held closely together, whereas here they separated slightly, they're spreading, as you can see in these two. So those are features that I only noticed that are not recorded in the literature, and I only noticed that they are very useful by engaging on iNaturalist. Unfortunately, on iNaturalist, there's a bias in terms of what people post, what they look out for. So you can see something very showy like this has got 227 observations, whereas a little minuscule, non-showy entity like Heliophila namakwana has only one observation at the moment. And there are several littlies like this with absolutely no observations because they are so diminutive. Then a lot of information can be derived from the growth form or habit. This is Heliophila lata, which, although it's a perennial with a woody base, it occurs in a group of annuals. It's the same Heliophila coronopifolia group, but it's clearly, you can't now just say because it's nested in that group that now one should call all these things Heliophila coronopifolia. This is clearly set about by its woody habit. Then habitat information is also important. This is Heliophila calosa, which occurs on Table Mountain and on southern slopes, which are normally quite bushy. So it's associated either with tall shrub land and has to compete with quite a lot of thick growth. So it has to be tall enough to emerge through the growth, and also occurs on forest margins, for instance in orange growth. Then Heliophila esterhuizeni is one of a group of woody plants like Heliophila calosa from the previous slide. So unlike the annuals, this has a proper woody stem, and you can see this from the siliques. They've got very large round seeds, whereas most of the other species have very flat seeds and are much smaller in diameter. So that, I mean, it's difficult to see the scale here, but these are probably about 4 centimeters long, those fruits. Then another group is the Heliophila merck-oweniana complex. The original one had white flowers, but if you go a little bit further south and north, you get to populations where they are. The populations could either be all white or all blue, or some blue individuals and some white individuals. So here it looks like this is more of a natural variable species or complex species which just contains a lot of variation. At a push, maybe the southernmost populations in the Oeverberg could be split off from the rest, but this just gives you an idea of some variation that one could expect even in one species. As you can see, it doesn't form a nice. It is the analysis didn't unite them together in a clade as in this group, but they all come out at one level in this cladogram, not resolved clearly from the rest. So, but this could just signify that it's an ancestral species, and that these there was a period of rapid radiation here, and then later these supported clades here diverged. This might be quite surprising that this is the closest extant relative of Heliophila, and it is very unusual in the proper leaves being totally different, but as well as the cotyledons, which are retained into maturity and function as green leaves. There are some other striking differences between Heliophila and Chamira. So just to show some other members of Brassicaceae other than Heliophila. This is Cardamine africana, which is a forest species, unusual for the family to grow in the shade in forest. And then the virtually cosmopolitan watercress, Nasturtium officinale, also common in South Africa. Then some invasive Brassicaceae. This is the wild radish, Urophanus raphanistrum, and Rapistrum rugosum, which can be easily identified by this these small siliques, short siliques with a globose portion in the upper half. And this also grows up to about 2 meters tall and really transforms the environment where it invades. Another quite radical transformer is Brassica tourneforti, which, just because with the sheer size of these leaves that lie flat on the ground, just shades out any other plant that could have germinated in that area. It has a very fast growth rate, so it outcompetes and shades out anything else. The threatened Brassicaceae species, there are a couple that have been where there hasn't been enough information, and they've been classed as data deficient, although some of these are likely to be vulnerable or have an even higher conservation status. In this case, Heliophila maraiana from Piketberg to the Gifberg is assessed as endangered, but this is doubtful, and since there are quite a lot of herbarium records, it doesn't seem to have any tremendous threats at the moment, so we might have to lower the conservation status of that one. Then a whole lot of species that are assessed as vulnerable, and the one in this area, the Swartberg, is Heliophila ephemera. Ishmael has taken some images of the type specimen, the isotype that we have in the Compton Herbarium, in case some of you want to go out and look for it. I hope the weather is good enough for you to be able to get to where you're likely to find this one. But yeah, as I said, there's still a lot to be reassessed in this lot. So a lot of species will be added to this list, and some might end up just being least concern. This one of them was this is one of the ones that is probably least concern, although it has some scattered, isolated populations. They're normally quite large, the populations, and it's got quite a wide distribution area. It seems to be one of these species that, when sea levels were lower, occurred right around the coast, and as the sea level rose again, it got isolated on the rocky promontories, and for where it's only known from today. Just some indication of the age of the radiation within Heliophila. Here's the closest extant relative, Chamira, that I showed you, and it got separated from Heliophila between 18 million and 12 million years ago. So at roughly between 12 and 10 million years ago, the Heliophila itself was divided up into predominantly Western Cape group, and then one that's either Western Cape or in the interior, going up into Namibia and into the summer rainfall area. As we progress in time, this group in the Western Cape was further split into the blue-flowered group that's so prevalent in the lowlands, and the more interior group on more heavier soils in the interior. And then as you go further, you can sort of trace, and some of this radiation is probably correlated with those fluctuating sea levels and fluctuating cold and dry versus warm and wet periods. But we are still in an early stage of trying to interpret the evolution of this group. INAT has been instrumental in bringing to light some new species discoveries. The first one that I've found was has now been described as Heliophila pseudo-eximia from the Richtersveld, discovered by Pieter van Wyk. Then Brian de Pree discovered this one. Well, I think it's also known from it was probably also known from the Bontebok Park in near Swellendam, but that still needs to be described as new. Then one of your local species, Wendy Wiles, was kind enough to send me some material. Unfortunately, not enough for a DNA sample, so we still need the proper DNA material for this. But it seems to be endemic to the Outeniqua range and just to the east of that. And this is a new species from the Toesberg, a perennial shrub, close to Heliophila florulenta. And this is an annual from the Karoopoort area, and at the moment we've got no idea where it slots in. So yeah, these are just 5 examples, but there are many more of new species that have come to light, and some of them specifically from our naturalists. Okay, so in the family, there are more new country records. There's the genus Lepidium, which, because it doesn't look like much, people don't really stop and look at it, photograph it. The flowers are minute, and the petals are hardly a millimeter long and greenish yellow, so not photogenic at all. But this is either just a new arrival in the country, or it's a new species in need of a name. Then, yeah, this is an example of one that is definitely a new species, probably related to Heliophila pusilla from the Oeverberg area. And Chris Whitehouse, being a botanist himself, he realized how important it is to peel back the sepals and peel back the petals and to show the nectary, and to show whether or not there are these appendages. In this case, the appendages are lacking, so that is further evidence that it's not one of the known species with which it could be confused. Yes, so yeah, I'd just like to conclude by recommending that people actually look for mature fruits as the first prize, but don't stop there. It's more the combination of characters that pins down a name for a species, and not just a flower or a fruit or a leaf on its own. So make sure to try and photograph as many different organs on the plant as you could. Or if you're preparing a specimen, to make sure to collect those parts, not only the flowers or fruits. The key in flora of southern Africa, and also the 2 volumes of Cape Plants by Manning and Snaman could be useful in getting closer to a species name. But it needs to be borne in mind that there are still there have been 16 more taxa described since then, since Marais at least, and that there are another 13 in the pipeline. So don't be too surprised if you don't find a name using the current resources. Then make use of indet to get closer to a name, and also discuss your thoughts on indet, or tag me and I will gladly lend my thoughts as well. Then I would really encourage people to submit pressed specimen collections of anything that seems interesting. And a lot of people just think, okay, well, this is just another blue-flowered Heliophila, so it can't be interesting. But especially in your area, the Southern Cape, any of these blue-flowered annuals would be very exciting, and could actually be new to science. So please feel free to contact me either via INET or via Crew or through SANBI, and I'll be glad to help you. Yeah, just a few more acknowledgements apart from all the INET contributors. I would like to acknowledge Crew, Botsok, the Grootbos Foundation for hosting us in the first workshop, and to that I can add De Hoek for this next workshop, the designers and developers of the Unnaturalist platform. Yeah, there's at the last count there were over 630 contributors of INAT observations. This is probably over 700 by now. I'd like to also acknowledge SANBI and international researchers of Heliophila. Thank you very much.